This figure is used to discuss how the gases are transported within the bloodstream. As you'll notice, it's similar to the previous drawing in that this represents the cardiovascular system, pulmonary circuit on top, systemic circuit on bottom. Notice this is where blood is getting its oxygen from the alveoli through external respiration. Down here is where the blood is receiving carbon dioxide via internal respiration. We already know how oxygen is primarily transported. 98.5% of the oxygen that diffuses into the bloodstream at pulmonary capillaries enters red blood cells and binds to or attaches to the iron within hemoglobin. 1.5% dissolves in the blood plasma. Once this oxygen is transported back to the heart and then pumped out of the heart and delivered to systemic capillaries, that oxygen has to be removed from the hemoglobin. So in upcoming slides, we're going to talk about how. How does that hemoglobin know that it's time to release oxygen? Why doesn't it release oxygen in the heart? Why does it know to wait till the right time until it's at systemic capillaries to release that oxygen? And it has to do with the changing of the affinity between oxygen and hemoglobin. Affinity is how tightly they stay together. How tight is oxygen attached to that iron? There are conditions in tissue that result in lowering the affinity. And lowering the affinity is a way that hemoglobin will know when it's time to unload the oxygen. Because oxygen has to be unloaded from hemoglobin in order to be able to diffuse out of the blood and then into tissue cells. So we'll talk about that on the next slide. But oxygen is transported mostly by hemoglobin, a small amount dissolved in plasma. Carbon dioxide, produced as a byproduct of aerobic cellular respiration, is a little bit different. A small amount of it is dissolved in plasma, but yet more than oxygen. So 7% of carbon dioxide dissolves in the plasma. 23% attaches to hemoglobin, but it's a little different. It doesn't attach to the iron, it actually attaches to the globin protein. And carbon dioxide also attaches to other proteins in the red blood cell. But 23% goes that way. But notice the majority. The majority, 70% of that carbon dioxide produced by tissue cells, undergoes a chemical reaction shown here in the box. Here's carbon dioxide. It's going to undergo a very simple chemical reaction. It combines with water. Okay, so there's water. Water's everywhere. This reaction is facilitated by an enzyme within the red blood cells. So this actually happens inside red blood cells. The enzyme is called carbonic anhydrase. What that does is it promotes this combination of carbon dioxide and water to form a very, very weak acid, but an acid nonetheless, known as carbonic acid. So all we did here is put these two together and we got carbonic acid. It doesn't look like we glued those together because we often put the hydrogens first, but there's 2 hydrogens from water. There's 1 oxygen here is from water, 2 oxygens from carbon dioxide, and the carbon from carbon dioxide. Now, by definition, acids dissociate. They release hydrogen ions. So that hydrogen ion is extremely important. One is it lowers the pH of this environment. 2, that hydrogen ion is one reason that hemoglobin releases oxygen. When this hemoglobin enters these systemic capillaries, these hydrogen ions bind to the hemoglobin and lower the affinity. And then hemoglobin releases that oxygen. So it's a way to know what cells need more oxygen. Cells that need more oxygen are producing a lot of carbon dioxide. So then where is the actual carbon dioxide atoms, the C, O, and the 2, or the 2 O's? It's found in this molecule here, HCO3 minus, and that is called bicarbonate. So 70% of carbon dioxide is found in this bicarbonate ion, and these things all get transported back to the pulmonary capillaries. Then this dissolved carbon dioxide, that's easy, it just diffuses out into the alveoli. The carbon dioxide gets detached from hemoglobin and diffuses into the alveoli. This guy's a little tricky. Bicarbonate is not a gas, so you can't breathe it out. That bicarbonate has to go the other way. So notice this reaction, the arrows can go either way. At pulmonary capillaries, that bicarbonate undergoes this direction, this reaction, to produce carbon dioxide again. And then the carbon dioxide is what diffuses into alveoli. So this is gas transport. We need to know the percentages of each. So what amount goes by what method. And this chemical reaction is extremely important for us because it tells us where most of that carbon dioxide goes and how it's transported. It also tells us that whenever you have a tissue cell or tissues that are producing carbon dioxide, that tissue is going to become acidic and produce hydrogen ions. Next topic, we're going to use this graph known as an oxygen hemoglobin dissociation curve. We're going to keep it fairly simple. This graph basically tells us that the more oxygen you have, this is the amount of oxygen, we call it partial pressure of oxygen. That's the amount of oxygen. This is the percent saturation. So that basically means how much oxygen is on hemoglobin. Is it 100%? Is it 50%? But you'll notice the more oxygen you have, the higher the saturation of hemoglobin. So this one's pretty easy. It just means the more oxygen you have, the more of it's going to bind to hemoglobin. So if you look at oxygenated blood, if oxygenated blood has 100 millimeters mercury of oxygen, that's going to saturate almost to 100%. So almost 100% of the hemoglobin is going to be full of oxygen. If you're down to deoxygenated blood, which is 40, about 75% of the hemoglobin is saturated. So you actually do have quite a bit of oxygen in deoxygenated blood. It's just not nearly as much as oxygenated blood. This graph is going to help us understand factors that affect the affinity of hemoglobin for oxygen. So when we're going to look at this curve, we're going to look at what are some things that shift the curve either to the left, which would be things that make the affinity greater. It increases the affinity. What conditions, what factors might shift this graph to the right? Those things lower the affinity. And we're going to focus more on the things that lower the affinity. You might want to remember that lowering the affinity is a way to unload the oxygen. And we want to unload the oxygen at systemic tissues. So the big things are A, acidity, or number one, I should say. The more acidic the environment, the lower the affinity. So hemoglobin will release oxygen. The more carbon dioxide, the lower the affinity. So carbon dioxide will actually promote the release of oxygen. The greater the temperature, so if the tissue is a little bit warmer, hemoglobin knows it's got to release more oxygen there. And then there's this molecule we'll talk more about in upcoming slides, BPG, or 2,3-bisphosphoglycerate. This molecule gets produced in response to certain hormones, and then that kind of knocks the oxygen off of hemoglobin. So we're going to look at each of these four separately on the next slides. So on this slide, you'll notice what we did with that curve. The curve from the previous slide is this black one. Notice that it can shift to the right when the pH drops, acidity. It'll shift to the left if the pH goes up, alkalinity, or a basic environment. So what does this mean? It means that if we increase the acidity, it'll enhance the unloading of oxygen from hemoglobin. This is a way that hemoglobin knows that there's a tissue that's metabolically active. It's generating a lot of carbon dioxide. It might be generating a lot of lactic acid, like contracting muscles. It's producing that bicarbonate and that carbonic acid. So metabolically active tissues need more oxygen. And the reason that acidity can do this is it's that hydrogen ion. The hydrogen ion in acidic environments binds to the hemoglobin, alters the shape of that hemoglobin, which will lower the affinity. And when you lower the affinity, it lets go of the oxygen. And that oxygen can be delivered and diffused into the tissue. This phenomenon is known as the Bohr effect. So the Bohr effect is when you have hemoglobin in a low pH, it releases oxygen. So if you look at the graph, we can say, let's just look at one of these numbers here, 50. If you've got 50 millimeters of mercury in a tissue that's 7.4 pH, the saturation is going to be over 70. So a lot of that oxygen is going to attach to the hemoglobin. If you lower the pH down to 7.2, notice the saturation drops. So that means less of the oxygen is going to want to be attached to the hemoglobin. So that's how you look at these curves. Again, we're looking more at these right shifts, because right shifts release the oxygen. This one, very similar. The only difference is we're not looking at pH, we're looking at the amount of carbon dioxide, which, remember, they're related. The more carbon dioxide you have, the more acidic the environment is. So increasing amounts of carbon dioxide enhances the unloading of oxygen from hemoglobin. Carbon dioxide also binds to hemoglobin, and when it does, it alters the shape, just like the hydrogen ions did from the previous slide, and that lowers the affinity. So I'll remind you of this chemical reaction. The more carbon dioxide you have, the more carbonic acid is produced, and the more acidic you are. So you're going to have more of these hydrogen ions and more of these carbon dioxide molecules in metabolically active tissues. So those tissues need more oxygen than maybe other tissues. For example, contracting skeletal muscle needs more oxygen than a tissue that's not very metabolically active. The next slide is very similar. The difference is we're looking at temperature. Tissue that is a little warmer, that heats up because of metabolism, will release more oxygen. So increasing temperatures enhance the unloading of oxygen from hemoglobin. Remember, heat is another byproduct of metabolism. Heat, carbon dioxide, and acidity are all results of metabolic reactions. A couple other factors. Here's that BPG molecule, 2,3-bisphosphoglycerate. It's a molecule that is produced inside of red blood cells in response to certain hormones. Notice all of these hormones are involved in increasing metabolism. Thyroxine, thyroid hormone, human growth hormone, epinephrine and norepinephrine during fight or flight responses, and testosterone. These hormones basically stimulate more release of oxygen because tissue that needs more metabolism, needs more energy production, requires more oxygen. So these hormones stimulate this molecule to be produced. This molecule helps to unload oxygen. So this graph could represent BPG, and that you would get that same rightward shift the more BPG present. This last thing is just a comparison. Hemoglobin is different in a fetus, in a fetal circulation, than adult. So fetal hemoglobin actually has a higher affinity for oxygen. This helps to transfer that oxygen from the mother's blood into the fetal circulation. It's a way to make sure the fetus is getting oxygen. If they were equal, same affinity, there would be equal amounts being distributed between mom's blood and the fetal blood. We want to shift a little more of that oxygen into the fetal circulation.